In exercise 5.2, we're going to explore the aerobic pathway by using lima beans, which are rich in mitochondria and have been processed, so we have a slurry of their mitochondria that were lysed when their cells were opened. We're going to combine that with a buffer. Recall that buffers help us to keep the pH at just the right condition, or near 7. We'll use DCPIP as a way to visually see the changes that are occurring during this aerobic pathway. And lastly, we'll be using succinate, which is our substrate. If we review the diagrams that are in the lab manual, you'll notice that the Krebs cycle, also known as the citric acid cycle, are the second stage in the aerobic pathway. In this cycle, which takes place inside the mitochondria, one of the steps is a conversion of succinate into a product called fumarate. Now, we don't expect you to know and understand every single step of the Krebs cycle, but that this critical step allows for the release of electrons and hydrogens. When those are released, things can pick them up, as in NADH and FADH2 could be carriers if this were a naturally occurring process. We are going to interrupt that process by inserting DCPIP, which has a high affinity for those hydrogens and electrons. So as these cells, these mitochondria, are going through the Krebs cycle, or the citric acid cycle, they'll be kicking out hydrogens and electrons, and the DCPIP will pick them up. So what will happen is the DCPIP by itself is bright blue, will attach those electrons and hydrogens to itself, and in doing so, it changes color. We should see a change from blue to lighter blues to total colorless, as more and more of the Krebs cycle is performed. Now, for this experiment, we're going to have 3 test tubes, each with the contents listed on table 5.3 on page 106 of your lab manual. Study this table. Note that we are using the same amount of mitochondrial suspension and DCPIP for each of the 3 test tubes. What will be different are the buffers and the succinate. Recall the buffers are there to correct the pH. Why do we have different amounts of buffer? Well, that's because we have different amounts of succinate. So as more succinate is added or less is added, we need to make sure that the buffering capacity of this solution is correct. The enzymes within the mitochondria are very sensitive, so we have to make sure we don't mess them up. We'll start with our mitochondrial suspension, of which we need 0.3 milliliters, which is a small amount, and that's why we use a very small pipette.